From Microbe TV, this is Immune, episode number 54, recorded on March 31st, 2021. I'm Vincent Racaniello, and you're listening to the podcast on the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi again. How are you?
Very well. How's the weather? You have cold or warm up there?
It's warm today. We are almost 60 degrees. And then it's going to snow again, and our ski resort is probably going to be open this weekend.
Are you serious? You're going to be able to ski?
Yeah.
Holy cow.
I probably won't, but yeah, it's insane.
It's very interesting. From Durham, North Carolina, Steph Langle.
Hi. Great to be here. Back again. Thanks all for doing a great job last month while I had some daycare shutdowns, but good to be back.
And from Madison, New Jersey, Brianne Barker.
Hi, great to see everyone. I'm excited to talk about another immunology paper today. Second one I've talked to Vincent about this week. So that'll be fun. And it's warm here too. It's almost 70.
Wow.
And
I was going to ask.
It's so weird.
Wow. It's warmer there than here.
Oh, really?
Yeah.
I figured you would be the warmest.
I mean, it's raining outside, I see, but...
Oh, it's raining and incredibly humid, but also 69 degrees.
Okay, well...
So you choose your poison when you want to complain about.
So, Steph, as you know, on a recent Twiz, we were talking about you and Linda.
I know. I heard that.
And I said, I said you were in Iowa, and so you corrected me. And I got an email from someone who said, no, they're not in Iowa.
Oh, that's so funny.
I said to Steph, it's all the same, isn't it?
Well, see, and I felt
New Yorker,
glad
right?
because when I heard you say that, I was like, man, I don't think so. But my reference...
Yeah, it's like,
But my reference was... I thought Stephanie was from a sports school, and I've talked about sports with her, so I thought it was Ohio State. But
Iowa people would be very mad at you.
I know, but I was specifically felt like I talked to you about Duke versus Ohio State sports.
Yes,
but that was my only point of reference.
Yes. Well, it's funny because when I almost didn't text you, Vincent, but I said, okay, this is my strategy. I'm going to text you how much I liked the episode, which I did, loved it. I'm going to say thanks for bringing up my, my past work, which was awesome. And then the next text I said, but by the way, it's another 4-letter state in the Midwest. It's Ohio.
That's a good strategy. Instead of saying you screwed up initially, which would be welcome with resistance, right? You were nice, and that softens the person up. What a brilliant career you're going to have.
Listen, I'm good with the people. Working with you. And I waited a week. I didn't wait till right after. So yeah, no, but it was a great episode. Well
trained. You're well trained. That's really cool. But yeah, we do have the New Yorkers view of the rest of the country. I'm sorry. I have no problem with New York State. I know where New Jersey is, but Iowa. Sorry about that.
No, it's fine.
I even went to Ohio not too long ago, right?
You did. Well, that's the thing for the Ohio State people. You did do a talk there. So, but both schools are great, of course. Good virology and immunology going on in both.
Well, Steph, in reward for missing last episode today, you get to do the paper.
Yes. So the paper that I'm going to be talking about today, I'll tell you the title, the authors, where they're from. So the title is Age-Dependent Reduction in Asthmatic Pathology Through Reprogramming of Post-Viral Inflammatory Responses. So there's a little bit in here for everyone in regards to, so we got the viral component. We're going to talk about macrophages and pediatric animal models, which I love. So the authors are, first author, Guy Hazan, and then the last author, Jeffrey Haspel, and a bunch in the middle. And all of these authors are from, I'm pretty sure all, WashU. Am I correct? Yeah, looking at it.
I think so, yeah.
All look like they're from Washington University School of Medicine in St. Louis. It's good to know where they are because in, I think, the first couple of paragraphs, they talk about some clinical data and they say in our area. I'm like, well, I don't know where your area is. So I had to look. St. Louis is what they're talking about. Okay, so this is in the Journal of Immunology, and I like the Journal of Immunology a lot. I like it like I like the Journal of Virology. These are association journals, and I think they bring us, it's not going to be like your jam-packed Cell Nature Science paper where there's 50 supplemental figures and it tells this huge full story, but it gives us some interesting interesting windows into immunological concepts. It also offers opportunities for us to talk about experimental design, what we liked, what we didn't like, and of course, as we do on these shows, all of this is said, and of course, we enjoyed the paper. But then, there's always things to improve. Yes. So we'll talk about that too.
I also love, Stephanie, that you mentioned sort of the scope, even though it is an 11-figure paper.
Well, I was going to ask, is this the longest JI paper that you've seen?
I kept going. I'm like, I'm surprised.
The longest JI paper I've seen.
I didn't know they allowed past 8 figures.
I thought there was a limitation.
Like a supplement. You could put that in supplementals. Yeah, I was surprised too. But, hey. A lot of this, there was something, I don't know what things I could have been supplemental, but that's a good point.
What did you think, Brian?
Yeah, go ahead,
Brian. I would agree. I think that there were a lot of really interesting observations made here that provided fruit for future follow-up. And I also saw some things that I thought probably could have been supplemental. I think this is the longest GI paper I've seen.
Yes. So what we're going to talk about today broadly is asthma, particularly pediatric asthma, and I'll define asthma. So their first sentence in their paper, asthma is a common lung disease characterized by paroxysms of airway obstruction, airway hyperresponsiveness, and inflammation. And if you haven't gone to medical school and you don't know what paroxysms are, it's a sudden reoccurrence or attack of a disease. So it's like a sudden worsening of symptoms, which is characteristic of asthma attacks, right? Your lungs, the lobules are constricting, and it's causing reduced blood flow and oxygen. And so this disease has a genetic susceptibility. And so there are individuals who have genetic contributions to asthma susceptibility, and that's has been shown through family studies, heritability studies, twin studies. And it also is exacerbated by things like infectious agents, viruses, like we'll talk about, allergens, and the allergens can range from pollen, mold, animal dander, insect parts, some chemicals. And it's also exacerbated through reoccurrent exposures. So exposures of these agents over time at certain age groups seem to exacerbate it. But what's interesting, and there was some work done at Ohio State, that in Amish communities, rural communities, farming communities, that the levels or the risk of asthma is actually lower in those children, particularly when mothers on farms, there is this hypothesis that low levels of recurrent antigen exposure, particularly endotoxins, could prime the system away from this, and we'll talk about what this means, a Th2-driven eosinophilic response in the lungs. And that's interesting, right? Because endotoxins are going to, it's LPS, that's E. coli, stimulate a different toll-like receptor on the surface of cells than something like a viral infection, an allergen. And so there are things that can reduce your risk of asthma. Having pets, being around livestock is one of them.
Yes.
I have a question.
Yeah.
So you said this really nicely, that when you have infections that drive more of a Th1 type of response, that makes logical sense of biasing away from Th2-driven diseases, and asthma is one of those. But what about, there's also the hygiene hypothesis where we've had better clean and water and so forth, so we've reduced our parasitic exposure, which is Th2, and that also seems to correlate with lower levels of Th2-driven diseases. And I've kind of always said, well, that's because they're teenagers that tend to get in trouble, and you occupy them with something like a parasite, they're not going to cause trouble another way. But the reality is, it seems like either one of those two could reduce the amount of Th2-driven disease pathology. And do you have thoughts on that?
Yeah. Oh, Brianne, you're going to say, you go ahead.
I've seen sort of two models that address this, and one of them kind of helps me understand this, is that in the case of the parasitic infection, you could imagine that in a population where there are many people who have chronic parasite infections, that the overall level of immune responsiveness might be a bit lower in the population, sort of the mean immune responsiveness. Everyone's a little immunosuppressed. And so there are fewer people at the high tail who are making very excess immune responses.
Excuse me.
While in a population that does not have that immunosuppressed phenotype, there are going to be more people who are sort of at the level that is enough to have an excess response. So I've seen some really nice kind of figures trying to depict that. And I think that that has helped me understand exactly the question that you pose.
Mm-hmm. Interesting. Do you also think it could relate to what I mentioned about lower levels of exposure early in life? And so the farm example, high endotoxin levels, LPS from animal feces or whatever, dog dander. Could that also explain that if you have if your response is generally type 2, that there's a reprogramming that there's not a hyper responsiveness. And I guess it could go back, Brian, to what you're saying, but that it seems to be in these cases where these pediatric patients or individuals, they're dipping into a Th2. It's like they're at a Th1 and then they can't handle the Th2 and it kind of accumulates upon itself to cause this attack. I don't know. I think maybe we could be saying the same thing, but something about, priming the immune system and keeping a general Th2 environment could be the reason. Sounds like we could be saying similar things.
We could be, yes. One other thing that I think was really interesting when I was thinking about some of the background information that they were discussing here is that they talk a little bit about differences in asthma with age and sort of what is seen in pediatric asthma and what's seen with asthma of different age groups. And, I am not a physician. I have not gone to medical school. But I think that I have always understood that adult onset asthma and pediatric asthma were different things and differed a bit in severity. And so I think that, and when people have asked me to go into more detail on that, I've said, well, yeah, I think that's how it goes. And I can hand wave some reasons immunologically why I think those would be different, but I haven't really seen solid data on that. Again, I could come up with a little mechanism if someone really wanted me to.
Get you a dry erase board.
Yeah, exactly. But I liked the idea of this starting to address sort of the idea of some of these disease states that are seen in different age populations may actually be functioning differently.
Yeah. Yeah. And actually, I'm glad you said that, functioning differently, because I think I do want to take a step back just a couple of minutes and reframe the way maybe we talk about Th2 type immunity, helminth infections. I think because I think immunosuppression is maybe the wrong picture of Th2, because I think it's more people who have more Th2 who maybe can better fight helminth infections. They actually, in some cases, not if they're HIV positive, are less likely to get cancer. There are other diseases that they're less likely to get than those who don't. So maybe a way to think about it is you just, depending on your environment, you're skewed either way, but one, type 2 actually could have benefits for this thing compared to this thing. So I think I hesitate with immunosuppression concept unless people are like on immunosuppressive drugs. If that makes sense.
Coming back to what you were saying, Brianne, about adults versus kids having different types of asthma, I did think that there's a lot of kids who actually have asthma that's triggered by various different things, but a lot of it, I thought, and I could have thought wrongly, a lot of it is infection-induced asthma, which is sort of related to this. So I think kids are frequently hospitalized after they're already sick and they develop bronchiolitis and some asthma-associated sequelae from that. Would you agree with that?
Yeah, I think that that's true. And I think that predisposition is specific to kids in many cases, that in some ways they sort of grow out of it.
And I think that's sort of what they're trying to address here is what might the viral infections be doing and how might that change over time? Like how might their response to the viral infection change over time, and that might change their predisposition to these asthmatic events. Yeah. And so that's why I was wondering, because if they're framing it this way, is that the same as someone who is not infected with anything that we know of having an acute asthma attack from some kind of, they breathed in something or we just don't know why, but they have this attack. Is that the same thing? And is that also impacted by this? That's sort of how I was thinking about this.
Yeah, I feel like that would be really hard to answer.
Yeah.
Because you would, if we think about some of these infections that we're talking about, many of them seem to be common, and you could imagine that in a lot of cases with that acute asthma attack, it would be hard to know if whether or not there had been a predisposing viral infection that maybe had gone undiagnosed earlier. So I think it would be pretty tricky to fully address that question. It's a really interesting question, though.
Yeah.
A vast majority of these are associated with rhinovirus infections. And often what happens is a kid gets infection, they start with a wheezing illness, which then progresses to asthma. What's interesting with the rhinovirus is that only certain types are associated with wheezing illness and asthma, types A and C, and the Bs don't do it, which is very interesting and not understood why. But so there's some virus component as well.
Interesting. And do rhinos infect mice? Is there a mouse model that you could study that?
It's funny that you should say that. We tried.
Oh, okay.
There is a mouse in the literature, but the virus doesn't reproduce. You can put virus into the respiratory tract, and it doesn't reproduce. But so I always thought years ago we tried to adapt the virus to mice, and it's funny because today I just got an email for someone who would like that virus to do some drug screens in mice, and it's like we threw it out years ago. Nobody wanted to work with it anymore.
Oh, no.
But I don't think there is a good mouse model. No.
Interesting. Okay.
But the thing is, the virus replicates very poorly, even in humans, and it doesn't kill cells. It doesn't kill cells. So some kind of sensing issue is going on with asthma, right?
So it's
People
all
are trying to
immune-induced pathology?
I bet it is.
Wow.
Yeah. Okay. Interesting. Well, Brianne and Cindy, what you guys are talking about is just a really great setup because they do give us some stats. And so, because they want to bring us into this phenomenon that they're roughly 50% of the children diagnosed with asthma before the age 6 will experience remission of their symptoms by early adulthood. And then 20 to 25% of the cases will achieve resolution of their airway hyper-responsiveness. And so the exacerbation rates really they fall as children age, and then they plateau when they reach adulthood. And so what is this? How is asthma aging out? And that's not understood. And one of the things that they talk about is that the seasonality of asthma exacerbations in children corresponds to viral disease. And so you can find that asthma in emergency department visits, they start when school starts. So they start in the fall and they peak during the seasonal flu, cold and flu, and then they go back down in the spring and summer. And so that's another indication that viruses probably play this large or maybe an outsized role particularly in children. And why is that? And one of the things that they present to us, so the first set of figures that they dive into is we have an experiment that COVID-19 gave us. and that experiment was what happens when we implement measures that do not allow for the transmission of childhood viruses. And that is COVID-19 associated lockdowns, which are masking, social distancing, shutting down schools, shutting down parks. And what does that do to the rates of emergency department visits due to severe asthma? And it's amazing. And there's a lot of anecdotal evidence that I had heard from my past postdoc mentor, Sally Permar, that they go away. That post-lockdown, the seasonality and the increase, the huge increase in emergency department visits in young children went away. I'm sorry, I shouldn't say that it went away, but the cases were the same. It's the same amount of visits per year as older age groups. And that's pretty incredible. I mean, it's a 70% reduction.
Yeah, and they also talk about how the seasonality, as you mentioned, also went away. So instead of seeing different numbers in different months of the year, that this sort of background number, the number they see in older and younger children, also was that same background number throughout different parts of the year.
Right. And so it implies this potentially developmental process that mutes the host response to seasonal triggers in children. And that one thing they mentioned is that and these were antiviral measures. And so I would like to tweak that, because masking, social distancing, while of course the end result is reduction in transmission of viruses, it doesn't remove the contributions from allergens. If we treated everyone with an antiviral, that would be direct evidence of an antiviral effect. I think that we can try to remember that these kids aren't going to schools, and schools have large amounts of allergens, right? Especially in older school buildings. There's mold, there's cockroach dander, so it wouldn't necessarily all be attributable to viruses, but for sure a large portion.
And one of the things that they said also was just the lockdowns in general reduced the overall respiratory virus positivity rate, right? So that's like one of those things, like everybody's saying, oh, the lockdowns don't matter, people are still getting sick, et cetera, et cetera. But I mean, their testing shows clearly that these measures actually reduce transmission, which makes perfect sense and is the basis behind health policies and things like that, even though people want to believe that's not true. It really is.
Yeah, I was excited that they cited their specific data on that, so that I could sort of see a data source. And I made note of some of the references that they actually used here in talking about that, to go back and take a look at some of them for my future use in classes, because I was in the same way being like, maybe these could be the perfect data to show this to those who need it to be shown.
What's interesting, though, is that as far as I remember, rhinovirus infections did not abate. I remember Amy saying this multiple times.
We looked at a paper on TWIV a while ago with some data on that.
Okay, that's interesting. I don't know what that suggests.
I don't either. I mean, she suggested maybe the transmission is different. And if you're locked down, you're staying home, right? So I don't know. But that would be a problem, because if we think rhinoviruses cause
driving
asthma,
this, yeah. And so I guess what you're saying, Vincent, and kind of a lot of this is, we're looking at broad trends and trying to speculate there's probably other contributions. And the other thing to mention, I mean, the brackets of time so the pre-lockdown was a 4-year average and the post-lockdown was 1 year. And so your confidence intervals in the pre-lockdown average are going to be tighter than the post, just because there's more kids. Right. And so you can even see that in the data where there are differences in emergency department visits. For example, in 6-year-olds, it's at 10 visits per year, and then it goes back up in 7-year-olds and then back down in 8 and 9-year-olds. So I think what this represents is it's not that there's probably biological relevance why your 6-year-old has less emergency department visits than your 7-year-old, but it's just there are less kids to make that mean more smooth.
That's in the allergy responses, which weren't different. And so, that's one thing that's really unique about this, is that the asthma visits have gone away, but the visits for other types of allergic responses really haven't changed, is how I was looking at this.
Yeah, it's interesting though, because again, I mean, those confidence intervals don't overlap. So I was wondering, it kind of looks real to me, but I don't know.
There really are not any confidence intervals on the 1 year post lockdown, because it's just 1 year, right?
Right.
So I think there are some caveats with this paper. It's provocative. It has some really interesting things, but I think there are some limitations.
Sure.
And that's one of them.
I think that in their favor on the asthma part, I agree with you completely on the allergy part, because I spent a lot of time squinting at that graph and looking at a couple of different points and thinking about what would happen if those points changed. On the asthma front, if we think about some of these lockdown measures, it's pretty clear. And have not been to St. Louis in a very long time. But I wouldn't say that all of the lockdown measures that happened over these years were 100%.
That is very true. It is definitely.
And with less than 100% mitigation measures, that's a huge difference in asthma case numbers.
Yes, you're right. You're right. So you're right. I think that's a great point. So they're saying maybe the allergic reactions are not different, but the asthma is. Hard to say about allergic reactions, but you can't argue with that asthma data.
Right.
So here's a little clarification on the rhino. I'm looking at a study where they summarize current non-pharmaceutical interventions were not effective in preventing rhinovirus infections of children, although they were effective against influenza and RSV. Rhino infections decreased during the initial lockdown with school closures, but as soon as the restrictions were eased, detections returned to near normal level, and this is in multiple countries. Unchanged rhinovirus detection rates during social restrictions.
Could that mean that rhinoviruses are even more fit to transmit?
Well, I don't know. It's a good question. I mean, one of the ideas is that there's more of a role for finger contact transmission than for SARS-CoV-2 and other viruses, right? And in fact, in transmission studies with volunteers and rhinoviruses, what they do is they put them in a room and they have one infected volunteer and they play cards, and the cards transmit the virus.
Interesting.
If they have one person sitting there who's not playing cards, so they're not touching anything, they don't get infected.
That's interesting. So potentially
not on cards.
Right.
Yeah. I remember when we looked at the data on TWIV, feeling like there was maybe something to enveloped versus non-enveloped viruses looking different, but I'm not sure that there was necessarily enough data to really make that as a claim.
Yeah.
Well, I think it's interesting because I don't know about you guys, but we don't have a mask mandate anymore, although it's still recommended, which you've probably seen in the news. Cornell makes news all the time now.
I know.
It's really great. But I am noticing that everyone is less worried about fixing their mask. So there's a lot of mask touches constantly pushing up,
right.
adjusting. And so even if you're wearing a mask, if you like you said, if it's more transmissible by touch and surface, then you still might get it more, touching eyes, wiping eyes, because I think very early on, even when we were being told the masks were working, it's less transmissible. Like people, if you remember from COVID-19, people were spraying down all their groceries and everything, every piece of mail. I mean, it was crazy, right? Then it was like, that's not the way you're getting it. You're getting it from breathing, so wear a mask. Right. So people were less worried about touching things. But yeah, I mean, the different transmission might be of RSV or rhinovirus might be related to that.
And I also think during lockdown, someone in the house went out at some point to get something.
Sure.
Right.
They get it on their fingers and they bring it home to their kids. And at home, they're probably not wearing masks, so they're touching their nose and so
wiping boogers off their kids.
I don't understand the asthma decrease. It must mean that there are multiple viruses involved.
Right. So I agree with you. I think that it maybe the contribution from rhinovirus also involves secondary viral infections. And they do talk about how having a pre-exposure to multiple viruses can contribute in particularly genetically predisposed individuals. So,
or it could be that, I mean, I agree. I think you could have multiple, or it could simply be you remove one and the others take its place, right?
Yeah.
Sure. Yeah. Right. So that was an interesting piece of data, and then some anecdotes that we've heard from colleagues, but they wanted to probe this further. And so what did we do? We built an animal model. We developed an animal model of asthma, or at least the manifestations of asthma, which in their case, they're modeling post-infectious lung remodeling. And so what that means is things called mucus cell metaplasia. And metaplasia is when cells pop up where they weren't before. And so these are mucus-producing cells, usually goblet cells. So you see an increase in mucus production from goblet cells after, in this post-lung remodeling. You would have an increase in type 2 inflammation based on inflammatory type 2 cytokines, IL-4, IL-13, IL-5, IL-33. And then they talk about M2 macrophages, which I'll touch upon in a bit. And then this airway hyperresponsiveness, which how they measure this in mice is they have this, I looked it up, but they have this system where they basically can intubate mice and inject methacholine. Is that the pronunciation? Am I saying that right?
I think so.
I believe
methacholine, and it is a parasympathetic chemical that binds to a receptor that causes you to cough, and it's a way of assessing the restriction in the lungs and like how quickly do your lungs respond to that chemical, and that's a measure of airway hyperresponsiveness. Now, when they show us their timeline, so what they wanted to do was they wanted to give the mice a viral infection, and they picked Sendai virus or murine parainfluenza virus. It's in the Paramyxoviridae family. And Vincent, I'd be interested to hear your thoughts on this, but from what I understand, that this is a virus that it persists long-term, and the lung pathology persists long-term, so they can model like this acute phase of infection, but then chronic lung changes are happening. They've picked day 49. They don't really tell us what's significant about day 49. I'm sure there were some other studies to demonstrate that day 49 is relevant. So it's a model of respiratory infection that can induce chronic lung changes.
Yeah, that's right.
Yep. And so they're showing us their model, and they're demonstrating that there's an airway mucus gene called, is it MUC5A or MUC5AC?
I always say MUC5A.
That's one of those things we were talking about before. That's one of those words you pronounce in your head, and then you're like, crap, I have to say this on a podcast, and I don't know how to pronounce it. So, okay, MUC5A, 5AC, which is a mucus-related gene. It's one way to measure it. It's higher in young mice compared to older mice. That was also true for this particular staining of mucus-associated glycoproteins. So they in the juvenile mice, there's higher levels of this PAS staining after the Sendai virus infection, but not in the mature mice. And so they're defining young and older mice as 6-week mice as the younger, and 11-month mice as the older mouse. And they are also looking at things like IL-13, which is our type 2 cytokine, and they see an increase at day 49. So this is more of a chronic time point, higher in the juvenile mice only after Sendai virus, and particularly seems dose dependent with higher levels of Sendai virus.
Yeah.
They look at
earlier. Earlier in the paper, they tried to tell us how these ages of the mice correlate to human ages. So they say that what was it? The 4-week-old mouse they say is equivalent to between a 2-year-old and 8-year-old human. They say a 16-month-old mouse is akin to late middle age in humans. I suppose one can make their own decision about what that means. And
didn't they say like up to, like right around 50?
Like 50, 40 to 50.
Wasn't that? I think they said that.
And then,
anyway.
I thought that was the citation.
Oh, maybe.
And then they also talk about 3-month-old, which they said is early adulthood.
Oh, that's funny that you say that, because I assumed that was the age range. I'm like, that's weird. A 2-year age range, but you're right. It's probably
the reference.
Because I was like middle old age. I don't know if it would
48 to 50 years old. Analogous to early adulthood in humans, 49. I thought it was like, that's really what they were saying. That's early adulthood in humans.
Good catch, Brianne.
Oh, that's funny.
So yes, they are kind of trying to model, and it's hard in all of these animal studies. So we try to do this in monkeys. We try to say, okay, an infant monkey, what does that represent as it ages in comparison to humans? And we can do this in mice, but of course it's hard because mice age very quick. I mean, they grow up very quickly. They're adults by 11 months of age. So take it with, whatever grain of salt you need for that. But for sure, they're trying to model the range over the course of the life of the mouse.
So I was thinking about this, and it's funny because I was just talking with people in my immunology class about how to look at data, and it's higher in one group than in the other, right? And so the question is, why is it higher? And one could say these mucus production and MUC5 and these cytokines and things are higher in the juvenile because they're over-responding. But one could also say that they're lower in the mature because they're failing to respond, right? Because we always talk about aging and how aging influences immune responses. And so it's saying the same thing, sort of, except it's really interpreting it a different way, right? Because they want us to believe that the juveniles are actually hyper responsive. But what is normo-responsive, right?
Yes.
Which one is high or low? Like, is there one in the middle, or does it flip at a certain point? And how does that relate to protection and pathology and things? I don't have an answer for that, but it's just an interesting way to think about it.
Yeah, but right. So, and I guess it is in the context of the question you're asking, because you're right, this hyper-responsiveness, at least some data that we're seeing with SARS-CoV-2, children have maybe higher levels of things like interferon than adults. And so in that context, that means better protection because that's clinically what we're seeing. So yeah, it is, you do have to, especially with these age studies, think about, okay, what is the context? And are we potentially like putting something on this age group without the data? You know what I'm saying? Like we're saying these are higher or lower, but is that really to what?
I mean, is this just because they're better at fighting the virus and this is an outcome of that?
Right. Which we will touch upon because there were some interesting statistical questions I had about some of that data. Because that's a good point. Exactly. What this figure is showing us is they want to show us that this chronic time point, day 49, where it's a post-acute infection, that there is this exacerbation in juveniles. But what happens in the acute phase? And so in a couple of figures, we'll learn a little bit more about that.
One thing is missing here is virus titers.
Yes.
Yep. Yes. They do look at that later, but they did not look that here.
In this experiment, they say the peak viral load is between days 3 and 5, but I would like to see that, because how do you know it was in your experiment, right?
Right. How do you know that they cleared at the same with the same kinetics?
Exactly. Yeah.
Right. And what we could do is we could jump to figure 5. And this is an opportunity of improvement that I would suggest to this paper if I was a reviewer, is that some of the data does jump around. So that question is a pertinent question, because if the juveniles just are less good, quote unquote, less effective at reducing viral titers, then that could lead to more lung damage, and then potentially you have more chronic effects. But so figure 5, they're showing us that after sendivirus infection, that the viral RNA levels are not different except for day 8, which they are. Don't you guys see that too? They say they weren't different at all, but they actually, that's different.
Well, it's at 8 and 12.
Right,
but
8 and 12.
I don't care about RNA. It's infectious virus that matters, right?
Correct.
RNA could be totally misleading.
That's right.
So
that's an opportunity for doing that would have been really helpful here.
I mean, you can't even say that it's too hard to do the infectivity assay, right? You don't have to do it at BSL-3 like you would have to do for SARS-CoV-2. You can do this, and there's no reason to do a PCR. Absolutely. This is totally uninformative, this panel B, because it could be pieces of RNA and slightly different pieces. So they'd need to look at infectivity.
Yes. And
they looked at the infection. They didn't look at infectious virus isolated, but they did look at how well it infects and the number of cells it infects in the mouse.
They did. And that was something interesting, that although, they say in the paper that viral RNA levels weren't different, even though they are, if you look at day 8 and 12. And maybe they didn't want to talk about it because the trend flipped. It went from higher in mature at day 8 and then higher in juveniles at day 12. And maybe they're like, maybe that's not biologically relevant. I don't know why that was not brought up. But they do show you that when they stain for cells in both the juvenile and mature animals, that there seem to be higher, increased amounts of SEV or sendivirus positive airway cells, that the frequency is higher. But they're showing us, of course, one representative image. I guess they do calculate it with kind of wide error bars. I could believe that, though. I think I believe the data. that the number of infected airway cells is higher, that could contribute. If you have more infected airway cells, you could have higher local damage.
Right.
Mm-hmm.
Right.
But they do suggest that they both clear the viral infection by 12 days. So that's helpful for their model, because then at 49 days, you shouldn't have any virus left.
Right. Right.
Easy enough to find out, right?
Right.
True.
Right.
There's little RNA and there's no more infected cells in the lung, but you're right. They should have done that.
Now that's probably reflected in the reviewers, probably immunology reviewers who wouldn't necessarily
want that plaque assay.
Yeah, it wouldn't have made it in Journal of Virology, but it made it in Journal of Immunology, because the immunologists don't care as much. They're like, PCR is fine.
Yeah, right.
And then I did have another question. So they did look at the weight of the animals, and they didn't see differences between these animals until they got to day 12, where there was more. Okay, so they defined the weight index it's the weight loss indexed to starting weight. So is this suggesting that a higher number means more weight loss or weight gain?
I thought it was weight gain, but
I also thought it was weight gain, because I expected the juveniles to gain weight as they aged.
Yes.
Right. So that's what we could, okay, great. I think it's just because the way that they state it in the figure is it says weight loss index to starting weight. But I agree with you. I think it's higher in juveniles, which we would expect. Juveniles are going to gain more weight because they're growing. So, yeah.
Yes.
Okay.
But they did say there's no difference, and there is a difference.
Right. So I was like, are you saying there's no difference during the acute phase, which is true, but in fact, there is a difference later on. So, and this would have benefited from non-infected controls. What are mice? What is their weight gain when you don't infect them?
Yeah. I mean, if you don't infect, if you have a 4 to 6 week old mouse, it's just going to go because they grow really fast.
Yeah.
So in this one, when they're infected, they go down first, and then once they start to clear the infection, they start to gain weight like they normally would.
Right. Right. So that we hopped down to kind of that figure because it wasn't included when talking about this late time point. So I guess what we could summarize from this first figure is, okay, maybe there's not huge differences in viral RNA, although debated, no plaque assay. There is higher amounts of infected cells, and then later at day 49, there's a clear difference in the amount of airway resistance, mucus gene expression, et cetera. So they next asked, well, is this phenomenon, can you see this in other viral infections? Because
can we say one more thing about this?
Sure.
Because I found this was really interesting.
Yeah.
So they were really careful to say, well, we checked male and female mice, right? Because that's always what everybody asks. Did you do it in male and female mice? Is it the same? And I even put quotes because they said the age effect is sex independent, which is true. So, there is an effect by age in males and there's an effect by age in females, but they completely didn't comment at all on the fact that the female mice have way, way higher levels of MUC5AC versus males. So clear, and I found this fascinating because a lot of Th2-driven diseases or also autoimmune diseases have a sex preference, and it's usually females. And I think, but I'm not 100% sure, and I should have looked this up, asthma is as well. Is there a female preference for asthma? Do you know?
I don't remember it being one of the big standouts, but I can only think of the ones that are like super dramatic, like lupus. So right. I think it's not that dramatic, but I think there is increase.
Yeah, lupus is a huge one, but also for viral infections, don't females tend to respond more robustly?
Yes.
And so that fits with this as well.
Yes.
So I was just surprised because it's not a little difference. It's a big difference.
Yes. Mm-hmm. Yes.
That the females are way more responsive. And so I just thought that was another, they didn't even comment on that. They just said the age effect is sex independent, which it is, there's a difference, but I just felt like
it would have been interesting. Yeah.
Here's one article which says in children boys have more asthma than girls, and in adults, women have more asthma than men.
Interesting.
Aha.
Interesting.
Okay. Well, this is
Well, they're just
young and old have more.
Are you looking at figure 2I? Is that what you're?
Yeah.
Yep, I see. Yep. Okay. Yeah, they're saying that the younger females have higher levels, but they don't have so here's what they don't have. They don't have uninfected young female mice.
Right.
So we don't know what the baseline levels are.
They don't have
They
any
left those
uninfected female mice. They only
There's
have
no uninfected female
They only
mice.
have uninfected male mice.
Right. So it's kind of like, well, that could just be, I don't know. It's hard to say what that means.
Right.
Yeah. But I do agree. We are seeing a lot of data on sex differences. And so there would be evidence for females having higher responsiveness.
Yep.
But you're right. That is something. So I did remember reading that now that the boys are higher
In
for pediatric
young.
asthma in young. So, okay. So then they wanted to say, well, what if, do we see this similar trend if we infect them with influenza? Mm-hmm. So they did kind of the same experiment, except their analysis of lung remodeling for influenza was at 21 days. And again, like it's kind of that's a good amount of time before 49 days. And so I'd just be interested in why 21. Is it because they didn't see differences at 49? So they're like, we'll do it at 21. But either way, they did see increased airway resistance in the juveniles, increased staining for glycoproteins, mucus-associated proteins, increase in MUC5AC. So, they're suggesting that this is also seen after influenza infection, but, there's a lot more that could have been probed. They did look at relative RNA expression, not infectious virus, and they only looked at it one day. So it's, I see what they're trying to do because they're trying to relate it to, one, a human pathogen, and two, is this, like, let's see if this is not just Sendai specific, that this is any virus. But I don't know if we can conclude that necessarily from this figure. And they don't really do much more with the influenza. So I guess it's to say at 21 days, juveniles have higher levels of airway resistance. We could say that.
Yeah. I think this was important to show that it's not a unique thing to Sendai virus, but it's more general to multiple different respiratory viruses.
That makes sense. Okay, so then they go into, the presentation, maybe I would have waited, and this could have maybe gone into supplemental, because they asked the question of if microbiome differences can impact allergy-related responses, immune responses in general. So if we wipe out the microbiota with a broad-spectrum antibiotic, do you see differences? And, the antibiotic did a great job at reducing the microbes, but it didn't change the differences. I mean, that you still saw increased airway resistance in the juvenile animals after Sendai virus infection compared to the mature mice. And they looked at gene expression, and they didn't see a change in that trend. So, I don't think it concludes that microbiota doesn't impact things, but I think if you wipe it out completely, the trend is still there.
Well, I think their data show the microbiome does have an effect, and they also don't talk about that. This is like another thing where the data are there, and they just kind of ignored it. So
now, do you mean?
So in the one figure, they're looking at the airway resistance. Okay. Uh-huh. And they plotted their control mice and their microbiome depleted mice on two separate graphs with different axes. And if you look, the microbiome depleted ones actually have higher airway resistance. So it's saying that they are hyper responsive. So just depleting the microbiome is changing.
Oh, I see what you're saying. Yeah, right. Like
the airway responsiveness.
I didn't catch that.
Even
I don't know if
in the PBS treated mice. Actually, I should say the PBS treated mice were both right around 6, but the infected juveniles were more like 14 in the microbiome depleted versus 8 in the control.
Yeah.
So to me, that's a difference, and they viewed it and analyzed it in a different way, and they kind of ignored that difference.
Yeah, I wonder though, with the error bars, if they would be different. I'm just trying to cross my eyes over.
It's hard when they're on different axes.
Yeah, why did they do
that? So my question is, do you think you could conclude from this that the microbiome effect is one effect, but there is a separate age effect?
Absolutely.
Yeah.
Because I think that, when we talked about this at the beginning, we mentioned hygiene hypothesis and the environmental exposures and things like that. And so, what I would take from this is it's not just those microbiome differences. There is something intrinsic to age, even if you erase the microbiome or make the microbiome the same. And so that's as far as I would have wanted to take these data, is just saying there's something besides the microbiome. Because I agree with you, the microbiome is clearly doing something.
Oh yeah, and more in one than in the other. The age related.
And I guess that's why they didn't graph it on the same graph, because they were asking the question of, did it remove the trend of the difference of seeing juveniles higher than mature, and not within the juveniles having a difference? Do you know what I mean?
I do. I mean, I guess,
because it's not relevant.
Argue all the little details, but like if you take the normal mice and you infect the juveniles, they have a little bit higher airway resistance than the wild types. But if you deplete the microbiome, the wild type, the mature, the older mice still have the same airway resistance, but the juveniles is much higher. So to me, that means the microbiome is having an effect on the juveniles. So they're trying to compare the difference between the adults and the juveniles in each case. But what I'm saying is, even juvenile to juvenile, there is a difference. So the microbiome is having an effect, and it's having more an effect in the young than in the old. But yeah, I don't know. I just feel like there's some things like that the way they chose to present the data, and they're making one conclusion for their model, they're ignoring a whole richness of the data, more information that's there.
Right. Like that's something that the differences are exacerbated when you remove the microbiota. And why is that?
And it's true, because they tried to argue that, if you, in the next figure, they're looking at MUC5A, IL-33, TREM20, TREM2, and they're saying that in each case, it's lower in the mature, it's lower in the mature, it's lower in the mature, it's lower in the mature. But if you look at control versus microbiome depleted, it's much higher in the juvenile. So the depleting the microbiome is having a huge effect, but it's not having an effect in the adults. And I think they're ignoring that. So,
and maybe
the microbiome in the juvenile is actually having a pretty significant effect.
So I think if I was the head of this lab, I have two projects for my next set of students, one about the microbiome and one about the sex differences.
Yes, definitely.
Absolutely.
Okay, well, so definitely more things to explore with the microbiome. And the next figure we so let's dive into, so what they wanted to then ask was, so they're looking at the different cell types that are in the bronchoalveolar lavages of these mice after Sendivirus infection in juvenile and mature mice. And they, there were differences. So for macrophages, there at day 8, there was a peak in juveniles, but not in the mature mice, and similar for lymphocytes, although the ranges were pretty wide, but that's something. They more focused, though, on the airway resistance increase in the juveniles, which we already talked about. That kind of decreases by day 12. So it peaks at day 5 and decreases by day 12. But I think looking at the macrophages, this data gives us a little clue into what's coming up next and some of these differences in the juveniles versus mature. So we'll keep that in mind, macrophages.
Yep.
They next want to look at cytokines. They say, okay, what are the different cytokines that maybe could define the divergence between mature and juveniles? And they look at bronchoalveolar lavages. And what they are, there's a whole panel of different cytokines, and they're using something called a PCA plot to look at these differences, which is principal component analysis. And it basically reduces the dimensionality of the data so you can easily see where the clusters are based on the treatment group. And they observed these different clusters. They observed ones that were differentially expressed in the juveniles, in the adults, ones that were overexpressed. And they were trying to ask, do cytokines correspond with airway disease? And so what they suggest is that the cytokines that are overexpressed in the juvenile mice are positively correlated with disease, but the cytokines that were overexpressed in adults were negatively correlated against disease. And the ones that were more likely to be overexpressed in the juveniles are things like IL-4, IL-5, and IL-13. Mm-hmm. So it's a similar story, I think, to what previous data was showing us, is that there's a drive towards Th2-based immune responses. And that this can have an effect post, in the chronic post-acute infection. Okay. A little cytokine data. Okay, so now what they want to talk about is, it's not just viruses that can potentially induce lung remodeling. It's also allergens. So they wanted to see if the same phenotype, where you have higher MUC5AC and higher levels of airway responsiveness and PAS staining, our mucus glycoproteins, did that happen in young mice after allergen trigger? And so the allergen they use is Alternaria alternata, and I didn't know what that was. So it's a fungus that develops on fruit in cold storage. So would be a common allergen if you have like a fruit bowl, for example, fruit in your house. I'm sorry, cold storage, I guess, would be in the fridge, but it's common on fruit. And so after they expose these mice to this allergen, they're looking at 10 days post-infection. So again, we're not consistently looking at the same time points in these mice in the lungs. And so I don't know how these models translate to each other. But what they're summarizing here is that there's a different type of response, that they still see induction of mucous cell metaplasia and type 2 cytokines in both juveniles and adults, but there was less eosinophils, less hyperresponsiveness in adults compared to juveniles. And so these are different responses compared to the antiviral, the viral-induced responses. And I think that's really all we can say. Because they don't take this allergen any further. There's not additional experiments. It's really just to suggest that allergens trigger different immune responses in this model of post-lung remodeling than viruses.
Yeah, I think that they would ideally like to be able to tie it back to that clinical data on allergens that they had earlier. Though we, as we mentioned, there are some questions on those data. Sure.
But yes, I think, right, that's a good point. They were trying to tie it back to clinical data because it isn't just about viruses. But so just another model of this lung remodeling.
Yeah, and I think that they also, at least at the very beginning, try to make the comment that the allergens are not seasonal in the way that some of the viruses are. As someone who suffers a lot from all of the trees in spring, I'm not sure I totally agree that allergens aren't seasonal, but
yeah, right.
I think that's what they're trying to get at.
There's what people refer to as indoor allergens and outdoor allergens, right? So the indoor allergens you typically can't get away from, and they're not really cyclical, whereas the outdoor allergens are cyclical. There's leaf mold in the fall, there's pollen in the spring, grass in the summer, and so depending on what you might be sensitive to.
So this is a very relevant paper for you, Brianne, is what you're saying.
Yeah, so depending on what you're sensitive to. I think it's important though, because if you're not like allergic to many different things, and we can come back to we were talking about how you pronounce the word for that, atopy, atopy, whatever. Before we got on, like if you're not allergic to a lot of things, if there's a cyclical exposure that every fall you get triggered from leaf mold, that may be very different than if you're continuously exposed to an allergen in your house, right? Because the continuous exposure and remodeling might lead to a different lung pathology, or potentially even tolerance over time. You might outgrow it or something. So I think it's an interesting thing to think about. Is it the allergen that's different, or is it the exposure, seasonality, or cyclic or chronic exposure?
right? So the next kind of sets of experiments they do is they do mass cytometry, which flow cytometry is how a lot of us typically identify cells, but mass cytometry is different in that you use metal isotopes to be able to detect different cell types, and mass cytometry has lower background. It can be ideal for image analysis. There's just a lot less background noise, and you can potentially have a lot more markers. And so they use mass cytometry, and they just want to see what is different in immune cells and the lung lavage, bronchoalveolar lavages of the juvenile and mature mice. And so I think there's three interesting things happening here. So the B cells there are similar frequencies of B cells in these animals pre-infection. They dip in the lungs during infection, and then after infection, the B cells in the mature mice are higher. They go back to baseline, but that's not true in the juvenile mice. They don't really talk about the B cells at all. I think that's interesting. That the juvenile mice, they do not the frequencies of B cells don't rebound. The other two things that are interesting, and they do comment more on this, I think, as if it's their model, is that the eosinophils substantially, well, I shouldn't say substantially. I mean, the percentage of CD45 positive T cells is like 1 to 3%, but they increase over the course of infection, which would agree with the phenotype of the animals, that they're more Th2 biased, leading to increase in eosinophils by day 49. But what they hone in on is alveolar macrophages, and alveolar macrophages are really important in immune responses in the lung. They can be two types. So there's M1s that are these classically activated macrophages. Then there's alternatively activated macrophages. M1 respond to microbial factors, the more Th1 pro-inflammatory. And then M2 are usually induced by Th2, and they undergo different types of metabolism and can induce collagen deposition and contribute to maybe damaged tissues. So they see that alveolar macrophages broadly, so they didn't define them here, but they're higher in the juveniles, both pre and post infection. They dip during infection in both animals, but they really hone in on alveolar macrophages as potentially mediating something here.
Yeah. So I wonder about this. So the way they showed this, again, is percent of CD45 positive cells. So if there's a new population of cells that comes into the lung, other things will go proportionally less. And the reason why I think that's important is because you said there are M1 and M2, which there are. There are also two different sources of macrophages. We've got alveolar macrophages and we have interstitial macrophages. And they're actually from different sources. So interstitial macrophages are developed from monocytes. So they get into the tissue and they differentiate into macrophages. The alveolar macrophages are embryonically derived. And I always get confused because I thought it was the other way around, but I'm 95% sure I'm right on that. Okay. And so those are not the ones that are being recruited. So how are their numbers changing? And are their numbers changing because they're actually going away and coming back? Or are their numbers changing because lots of PMNs come in and then lots of PMNs go out?
Which you see in this data. Like you see, right. Now it's not the same magnitude in both juveniles and mature. There are a lot more Correct. PMNs, which is polymorphic neutrophils, that yes. come in the mature, in the older animals. Correct. But you're right. When you're looking at the percentage of CD45 positive, it's highly sensitive to changes in cells as they move in and out because it's a frequency. So if you have more of one, you have less of another.
Right.
Yeah. So if they could show us absolute numbers, which of course would be challenging here, that would be nice in addition to showing us the percentages.
Mm-hmm. Mm-hmm.
Right. Yeah. So, it took them down a road of trying to understand maybe the contribution to alveolar macrophages in this hyperresponsiveness phenotype, chronic lung remodeling after exposure to Sendai virus. And they do bulk RNA-seq and they do single cell RNA-seq. And what they pull out is that there's differences in MHC class 2. And so MHC class 2, it's going to be responsible for presenting processed antigens. It's to CD4 T cells and to stimulate that antigen-specific response, and obviously critical to initiation of the response after infection. And so they demonstrate that MHC expression increases with age overall. So it can be induced after viral infection, but overall it increases. But what was interesting is that at least in the one assay, now I don't work in this field, so like you'd have to tell me if, they probably could have done more to demonstrate that antigen processing and presenting was actually different, but they say that there's no difference in the antigen presentation, even though the expression is higher in the mature animals.
What do you think, Brianne?
I think.
Look at these bars. I mean, I don't know. I don't even know what to, because they are different, actually.
They are different, and they say there's no difference.
They're different. Here's where they're different.
They even have a statistic there.
There's a stat there. Yeah.
Their stats. I have some issue with it, but they say there's no difference, and they show there is a difference, according to their own analysis.
I was going to say, which would, I don't know if it's because it contradicts. So the juvenile is in alveolar macrophages that express what they're saying lower levels of MHC2. Mm-hmm. They seem to have a higher proportion of this. This assay uses ovalbumin to show presentation of this processed antigen. And it is higher in the juveniles, which I don't know. I don't know what you say about that, because these are alveolar macrophages that express MHC at lower amounts. When you get to the column that says, okay, alveolar macrophages expressing high amounts of MHC2, maybe they're not different. Juveniles still seem a little higher.
Is this more about the triggerability of those cells? I don't know.
Right. So, I think their transcriptomic data, I think that's probably more clear. I mean, they do pathway analysis, and they're saying that, okay, the pathways and the genes associated, they do seem to be different in mature versus juvenile. But I don't really necessarily know if it contributes to anything other than it's just different in age groups.
Yeah, I'm not sure. I don't think that this assay that they were using measures antigen presentation. Because as my understanding is that DQ ova is highly labeled and is quenched. And so when it's internalized and degraded, it now fluoresces. And what they did was they intranasally injected DQ ova into mice, and then they're looking at lung. So this means that whatever they instilled was taken up by some cells and broken. The ovas started to be chewed up, right? Mm-hmm. In some compartment where it's, endocytosed. And so presumably those chewed up pieces would eventually go on to an MHC class 2, right? But they didn't measure ova getting onto MHC class 2. And there are reagents to look at that if they have the right mouse strain, but they didn't look at that.
Right.
Because you can measure MHC with ova peptide in it with a specific antibody.
Yep.
As long as you have the right mouse strain. But they didn't do that. They just looked at, did cells take it up and start to chew up ova? But most cells will take it up and eventually start to process it, because there's a lot of proteolytic enzymes in endosomes. So they're MHC class 2 positive alveolar macrophages. But I think this just tells us that, at least in the juveniles, the high and lows take it up equivalently and have equal ability to process, to chew up the ova. Whereas the more mature ones maybe either take it up less well or chew it up less well. So they have less proteolytic capacity, which kind of fits with other studies that have kind of shown that the aged mice have defects in phagocytosis, in microbial killing, in antigen processing, et cetera.
I think that's a really great point. I like how it ties in with what you had mentioned earlier about that question of, are we showing that juveniles are making excess responses, or are we showing that mature individuals are making decreased responses?
Right.
Which one are we considering as the normal?
I don't know.
Right.
It depends on perspective, right? There's something, what was it? So when you're looking down, and the other person is looking down, there's something that you either see one thing or the other thing, depending on which perspective you're looking at it in. There's some kind of meme that I'm
not thinking of it, but if you send it, you should email it out and then maybe we can figure it out. So I think overall what they're trying to find differences and they're suggesting, okay, even looking at single cell transcriptomic data, that it's like these alveolar macrophages, they have more Ki67 in the juveniles, which is a proliferation marker. And so, okay, they may be important in juveniles. And so what's more interesting, I think, is that if you were to pull out these alveolar macrophages, so there are more of them in juveniles, or at least, okay, the percentage is, the frequency of them is different. And if there are a higher proportion of them, maybe that's driving some of this. Are they M2? Are they driving Th2? Is that explaining the phenotype? I actually think that there's really interesting things to look at in regards to alveolar macrophages that could explain this model, but was not explored. It's like a lot of things left over to look at.
Well, I look at data like these single cell RNA-seq, and I keep saying this, like every paper we look at, it's like they did this experiment to ask one question. And there is just this incredible amount of data that is associated with that particular type of experiment that just goes and gets deposited somewhere and people move on to the next thing. So if you are a trainee, if you can figure out how to analyze single cell RNA-seq data well, there is just a lifetime supply of data that have already been deposited with new stuff being deposited every day that can be just mined for amazing types of questions that nobody's thinking of, but they're out there.
Right?
Yeah. And as you say that, and I think related to a number of other things we've said throughout this paper, if any of the graduate students, current or future, from this lab are listening to this episode, we're sorry for the to-do list that we're making for you.
You can each have your own project and go start your own lab. There's plenty to do.
Plenty to do. So, we could talk about the last experiment because they've highlighted these alveolar macrophages as something might be going on in the juvenile. So let's knock them down in the mature mice. Now, I had to look this up. I've never depleted macrophages from mice.
Yeah.
Well, no, have I ever? No, I've never depleted macrophages from mice. Macs aren't my main area. So they say that so clodronate liposomes. This is what, have you used this, Cindy?
I didn't do the experiment myself, which is amazing, but I've read lots about it. And so what happens here is you basically take little liposomes. So that's just little lipid balls, and macrophages love to eat these things. They just gobble them up. And what you do is you load them with a drug that'll kill the macrophage. So the macrophages that gobble these up, they get killed. And so other cells don't really gobble up these lipids.
So they don't. So there are other phagocytes and they don't like these things?
So neutrophils will probably eat them up, but they're not really looking at those. They're just going to ignore that.
I was wondering about this method because when I looked up, it just seems like a phagocyte reduction method.
It is a phagocyte reduction. However, neutrophils turn over so fast. You're really depleting macrophages, and the macrophages don't come back very quickly. But I mean, neutrophils are constantly being put out of the bone marrow, so they'll just rebound pretty quickly. Okay. So if they're in the lung, they get depleted, they'll just come right back. But the macrophages, if you deplete them, it takes them a while to come back.
So I have a question, and I will completely admit that this question is based on the paper that we talked about on TWIV earlier this week that I still am thinking about, and I still have 100 experiments I want to do and just think it was the best paper ever or most interesting paper ever. Does the lipid matter in terms of which lipid you use?
I don't know.
Lipids matter for mRNA vaccines, don't they?
They matter for a lot of other things.
They do, and they matter in mouse versus human, and IL-1 in IL-1 beta and IL-1 alpha and IL-1 RA production, and you should read that paper.
Oh,
okay.
Or I'll listen to the TWIV and on my drive.
Both, yeah.
Yeah, both, yeah.
Yeah, I think it's just, people say clodronate liposomes, and I think it's just a pretty standard approach that will theoretically deplete the macrophages.
Okay.
And there's a cool thing that you can do too, because I mentioned those interstitial macrophages, which are in the tissue in the lung, versus the alveolar ones, which are actually hanging out in the lumen of the lung. So if you instill clodronate liposomes, it'll deplete the alveolar ones, but leave the interstitial ones. If you inject IV with clodronate liposomes, the blood will reach the interstitial macrophages, and you'll deplete them, and you can leave the alveolar ones. So you can do experiments to try and differentiate which cell population is contributing, which macrophage population is contributing to phenotypes that you see in the lung.
Cool.
Yeah.
Do we know, I'm looking back in the methods, do we know if they infuse them?
Killed them in the nose. So they are depleted in the
I see,
alveolar
yes.
macrophages. Yeah.
Got it. Okay. Okay, so depleting alveolar macrophages, and after that, looking to see if there are differences. And so it seems that it did deplete them, so they were successful in doing that. And so the mature animals had higher amounts after clodronate liposomal treatment of our PAS staining, which stains for the mucoglycoproteins. Higher levels in the mature animals now compared to juvenile, which it was always flipped before. And this is at day 49. And that the airway resistance after the treatment, it still is significantly higher in the juveniles, and they don't take it out to day 49. So I'm like, I guess, I don't know what exactly they were trying to show here, but it didn't really flip the difference.
Nope.
No.
I think what they're showing is that in the control liposomes, so that the macrophages are not depleted, the alveolar macs are not depleted, you have a very high airway resistance in the juveniles and a lower resistance in the mature. But if you deplete the macrophages, the juveniles stay the same, but the mature ones come up and look more like the juveniles. So if you get rid of the macrophages, the mature ones are behaving more like the juveniles. That's what I saw in the airway hyperresponsiveness.
Mm-hmm. Yeah, similar, right? Like it's not that the trend flips, but it's that they come up.
Closer.
It brings them closer. Yeah.
Yeah.
So that was figure 11, and you were talking about many figures. That's a lot of data. And so to summarize, what we saw after COVID-19 lockdowns was that the amount of kids showing up in the emergency department for asthma was significantly reduced compared to pre-COVID lockdown years. Building this mouse model is relevant if we want to try to test some of these hypotheses regarding viral induction triggering triggering lung remodeling after, I guess, 49 days in this model. Depending on the virus you use, it could be different, but in a chronic setting. Whether alveolar macrophages contribute to this, I think is still up in the air. I think they do something, but I don't necessarily think that it was conclusive to show that they didn't demonstrate that they were the reason why you see these type 2 responses with higher airway responsiveness in the juveniles. Mm-hmm. But they do change as the animal gets older. Yep. So it's probably one of the things that contributes.
Yeah, I think macrophages are protecting the older individuals from the airway hyperresponsiveness. So
and that in and of itself is cool.
Then they get more. Yeah, I thought so.
Yeah. And why that is, what is it about them? I think more experiments.
I think that part of what they're trying to say is that they're functionally different.
Right. They talked about the reprogramming.
Right. Mm-hmm. So something about in the older individuals, those macrophages are better at repairing and quieting, whereas in the younger mice, they're more, I guess, inflammatory, or maybe so it doesn't matter. I don't know, maybe that wasn't the way to say it, but in the older mice, they're more important. The macrophages play a more important role So if you deplete them, now you're worse off.
Right. So that in itself is interesting.
Yeah.
I think the idea, again, that we can have some data showing how these phenotypes in different age groups are, in fact, perhaps mechanistically different is a really good thing. That it's not just sort of this anecdote of the asthma in the younger individuals and the asthma in the older individuals seem kind of different. But actually starting to move towards what is different about them is an important thing to do. And there's no shortage of additional questions to ask.
No.
Yeah, for sure. Well, good. Well, anything else about asthma that we want to say? Any wrap-up comments?
So the use of this model is a little weird, in my opinion, because this is not what happens in people, right? You have this chronic lung, you have an acute infection, it comes and goes, and multiple ones of those somehow predisposes. So I would rather they did that kind of model, right?
Yeah. So, right, like having a mouse model that has more acute.
Yeah, an acute
damage.
infection without this, long-term lung injury. I mean, it seems to me that that's not what we know is happening in kids, right? So why would you do that as a model? You're going to learn different things that may not apply to people.
Right. I think there's advantages to using various different models. I think the disadvantage of this is that you have mice that, they're not perfectly sterile, but they're specific pathogen free, right? So they have a pretty naive immune system, and you're giving them one insult, waiting, and then looking. And like I said, that's not really what happens. We're constantly being bombarded by things that we breathe in, especially when you're young. It's new exposures all the time. We're generating immune responses to it. And if you're talking about viral infections, respiratory viral infections, you're going to have one, and then you're going to get mostly better, and you're going to have another one. And then you're going to get mostly better, and you're going to have another one for your first couple of years. I mean, you know this, Steph. I had kids too. The first 2-3 years of you're constantly sick because they're constantly sick. I think every time they start to get better, they come down with something else. And so it's not a one and done. It's a repeated exposure. And so it's what happens in between those, and as you're piling on another one and another one, how is that repair process happening? And if you're constantly having a high pathology response, you're going to remodel the lung in a way that's going to have a long-term consequence.
And so I think that, Vincent, I do hear what you're saying, and I see what you mean about wanting more acute measurements. But I think to what Cindy's saying, okay, so what we're learning is that, particularly if you have this Th2 predisposition, after a viral infection, you have lung remodeling. So if you're hit again, it's probably going to be worse. And that, they didn't do the second hit, but we could assume if there's more damage that is what causes piling on of the Th2 immune response to cause asthma. I guess I'm assuming there's probably not good mouse asthma attack models that, that recapitulate what we see in humans. But taking this model and doing a reinfection would be really interesting to try to model that recurrent exposure.
And also that's not taking into account bacterial infections, right? Because the viral infections predisposed to the bacterial infection. You have pneumonia. There's massive lung remodeling. How does that impact all of this?
Yeah.
Because the kids that go to the hospital are probably not having a mild case, right? They're probably having a significant event in response to the viral infection and potentially developing pneumonia. Or I don't know, what is the trajectory versus developing an asthma versus pneumonia? And how do you distinguish that? Because it starts off, like you said, with a wheezing, right? There's a wheezing and then a bronchiolitis and then trouble breathing. And is it just because they do an x-ray and in one case you see lung involvement and you say, yes, there's a pneumonia, either viral or bacterial, or there's not? How do you make those distinctions between the asthma and pneumonia in those children that come into the emergency department? I don't know.
Should get a clinician on.
Yeah.
So we are doing a paper on TWIV tomorrow, which is kind of related. It's in Cell Reports. It is asthmatic bronchial smooth muscle increases rhinovirus replication within the bronchial epithelium.
Very relevant.
So rhinovirus infection is implicated in the vast majority of severe asthma exacerbations. And apparently the susceptibility of the epithelium to rhino infection is increased in people with asthma, so they're trying to get at the mechanism. Mm-hmm. And what they find is that these asthmatic bronchial smooth muscle cells downregulate the PKR pathway within the epithelium. So
interesting.
Yeah.
That is cool.
It's not even the target, right? It's a week of asthma.
Yeah.
We should coordinate and have like themes,
themed weeks.
We should, yeah.
Yeah. Well, thank you, Steph.
Yeah. Thanks, guys.
That was fun.
Very cool.
This is really
cool. Despite the the criticisms, it still makes you think, and it's useful.
Well, I think it's good for trainees when we walk through these papers, because it's these are all good things to talk about for experimental design, and what would you do next, and what would you rather do, and so
Yeah, and I don't think that anything that I was saying was necessarily criticizing what they showed. It's what they showed and didn't talk about.
I know, it's kind of like
interesting and important.
Yeah, it's like, oh, look at that significant data, but like
you just ignored it.
Yeah, I know.
Or it was like, it's not significant. You're like, what? It is.
Right.
Exactly.
So, I think what we're saying is just that the 11 figures weren't enough.
Well, that's what I'm thinking. You guys want more experiments, and now
I know, we're so mean.
It's going to be even bigger.
Well, yeah, you wonder, could that have been more than one paper?
Well,
where
yes, that's
it was
right.
smaller and you had more information about the data that were actually shown.
Right. Yeah.
I don't know.
Sure.
I don't know, because I can understand why they kind of will put a little criticism, if this is you who was on this. I think they did a bit of a notebook dump, because you do a giant single cell RNA-seq experiment. It doesn't tell you much. Where are you going to put it? You got to put it somewhere.
They want to publish it, yeah.
Right? You did an RNA-seq experiment. You did, cytoph and whatever. It's like huge, expensive experiments that showed a little bit. There were a couple of things that you could pull out of it. That's not an easy thing to put together.
Yeah.
No, it's a problem. Yeah. People want to publish it. Yeah,
I can relate to that.
And it deserves to be published. Like, even negative data are important to publish so that somebody else doesn't waste all their time doing it, right?
Mm-hmm. Yes. Agreed. Yeah. All right, that is Immune 54. You can find the notes at microbe.tv/immune. If you have questions or comments, you can send them to immune@microbe.tv. And if you like what we do, consider supporting our efforts. You can go over to microbe.tv/contribute. And we are a 501c3, so your contributions are federal U.S. tax deductible. Cindy Leifer is at Cornell University. Cindy Leifer on the Twitter. Thanks, Cindy.
Thank you.
Steph Langle's at Duke University, which is not in Iowa or Ohio. Stephanie Langle on Twitter. Thanks, Steph.
Yeah, thank you. This was fun.
And Brian Barker's at Drew University. BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
I'm Vincent Racaniello. You can find me at virology.ws. Music on Immune is by Steve Neal. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.